Hi, my name is Kim, and my presentation is on lymphoma and genetic sequencing. The lymphatic system is part of the circulatory system that consists of lymph nodes and vessels that move lymph fluid throughout the body. Lymph fluids contain white blood cells that fight infection. Lymph nodes filter and eliminate bacteria and viruses from the body. Lymphoma is a type of blood cancer that originates in the lymph nodes, and it metastasizes by spreading abnormal white blood cells to other parts of the body. There are two types of lymphoma: Hodgkin's lymphoma, which contains Reed-Sternberg cells, and you can see a picture of this on the right. These are large abnormal lymphocytes that may contain more than one nucleus. And then there's also non-Hodgkin lymphoma, which do not have Reed-Sternberg cells. Both types of lymphoma have 4 stages. Stage 1 is cancer in one lymph node or one organ site. Stage 2 is when cancer is in 2 lymph nodes near to one another and on the same side of the body, or the cancer is in one organ and nearby lymph nodes. Stage 3, at this point, cancer is in lymph nodes on both sides of the body and in multiple lymph nodes. Stage 4, the cancer can be in an organ and spread beyond nearby lymph nodes. As non-Hodgkin lymphoma progresses, it may begin to spread. The most common sites for advanced non-Hodgkin's lymphoma include the liver, bone marrow, and lungs. There are 2 kinds of biopsy. The first one is on the top, which is a traditional biopsy. This is an invasive procedure that removes cells from a suspiciously enlarged lymph node. You can see a picture of this to the right. The other type is something known as liquid biopsy, and it involves taking a blood sample, and it's to obtain circulating tumor DNA. As you can see on the picture on the bottom right, it's a lot less invasive. According to Dr. Maximilian Dean, circulating tumor DNA has exploded onto the scene in clinical care. When someone has a tumor, there are dying cells within that tumor, and its DNA gets into the blood. This allows medical personnel to get access to the DNA with a simple blood draw. There is a very small amount of DNA, so you need very sensitive methods to detect it. Also, according to Dr. Ash Alizadeh, it does take a soberingly long time to find out if a cancer treatment is effective, and the idea that we can find out that much earlier using DNA has been quite compelling. This not only provides information if a treatment is failing, but it might pick the next therapy that might fix that failure. After a patient receives radiation or surgery to cure that cancer, we can find out if patients are likely cured who can choose to skip the chemotherapy that occurs after surgery or radiation versus those patients that still have microscopic cancer cells would be ideal candidates for a more aggressive treatment. The more far-reaching application would be using circulating tumor DNA to find mutant DNA for early cancer detection before they spread, or cancer screening when there's still a high chance for curing them. Every tumor has specific genetic profiles that require specific types of therapy. Heterogenic tumors are a major obstacle to avoid relapse and succeed in remission. This is theorized to be an evolutionary survival tactic of metastatic tumors. If any number of tumor cells remain circulating in the patient's body after treatment, the chances of relapse increase. Single-cell sequencing has the potential to eliminate all cancerous cells, unlike the spatial bias limitations of biopsy. This is because single-cell sequencing has the potential to detect every type of tumor that requires quote-unquote precision medicine for every cancer cell in the tumor. The process of single-cell sequencing has its limits though. there is a currently higher error rate and misreads or mismatches in base pairs. Separating single cells for assessment of its genetic components is very time-consuming, expensive, and labor-intensive. The only way to supply the high demand of every cancer patient is for technology to advance with increasing identification sensitivity with increased accuracy rates and speed to accommodate every patient who would benefit from this. Pharmacology must also keep pace with the advancement of genome aspects of healthcare. The percentage of circulating tumor DNA in the blood is dependent on the lymphoma subtype. A study that assessed circulating tumor DNA in different types of lymphomas found that mediastinal gray zone lymphomas and primary mediastinal B-cell lymphomas had the highest levels of circulating tumor DNA. Diffuse large B-cell lymphoma and transformed follicular lymphoma had lower amounts, but still is considered to be high levels of circulating tumor DNA. Classical Hodgkin lymphoma and follicular lymphoma had the lowest levels. In a study conducted using 80 new diagnosed patients and 32 relapsed or refractory classical Hodgkin lymphoma patients, CAP sequencing, which is a form of next-generation sequencing, was used to assess the number of abnormal circulating tumor DNA compared with micro-dissected tumor biopsy specimens. Liquid biopsy found 106 mutations compared to 96 mutations found using biopsy. 84 of the 96 mutations found in classic biopsy were found using liquid biopsy. Another study involving 24 patients with advanced stage classic Hodgkin lymphoma had plasma samples evaluated during therapy. A 2-log decrease in circulating tumor DNA after 2 cycles of radiation treatment correlated with a complete response to treatment. A decrease of less than 2 logs of circulating tumor DNA correlated with progression of the disease and a higher mortality rate in classic Hodgkin lymphoma and also with diffuse large cell B lymphoma. 6 out of the 24 classic Hodgkin lymphoma patients had progression of the disease after treatment. 4 out of the 6 patients with progression had false negative scans with interim PET-CT assessment. All 6 of the patients with progressive lymphoma failed to achieve a 2-log decrease in circulating tumor DNA. This data suggests that genotyping using circulating tumor DNA may have relevance in assessing the status of lymphoma patients while undergoing treatment, which can aid in tailoring the proper treatment for the individual patient. Liquid biopsy shows patients that undergo non-invasive assessment. The sensitivity and specificity in single-cell sequencing must continue to progress to become a dependable method for customizing treatment for individual Hodgkin lymphoma patients. Next-generation sequencing is a DNA sequencing technology that has revolutionized genomic research. Sanger was the previous sequencing technology that took over a decade to decipher the entire human genome. Next-generation sequencing can sequence the entire genome within 1 day and has the capability to capture a broader spectrum of mutation versus Sanger. Sanger is restricted to discovering only substitutions and small insertions and deletions, and the next generation sequencing is unselective and interrogates full genomes or exomes to find new discoveries of mutations and disease-causing genes. For now, technology needs to continue to advance to increase specificity and sensitivity to detect all circulating tumor DNA. For now, it's being used primarily for research. Next-generation sequencing has optimal sensitivity to detect low amounts of variances of cancer-causing genes, which can provide a more accurate diagnosis, prognosis, and providing a customized treatment for cancer patients. Unfortunately, there are resource limitations such as computer capacity and storage, as well as a limited number of experts who can assess the data next generation supplies. Thank you for your time.